Hello, welcome to our problem worksheet on biotechnology. Let's take a look at a few problems regarding this material. Please make sure you also pay close attention to the lecture because there's quite a bit of vocabulary that would not be covered in this problem worksheet. So our first problem, we're trying to make some recombinant DNA. Oftentimes, one will try to put a piece of some DNA that we're interested in this case, Arabidopsis, into a vector. That vector can then be taken up into an organism. More copies can be made. It can be expressed to do new things. Lots of possibilities. So in this case, we have a cloning vector and Arabidopsis DNA. And we want to make a recombinant plasmid by putting some of this Arabidopsis DNA into the vector so that we can use the blue-white screening technique in order to determine the products that have the recombinant plasmid. You may remember from the lecture that the blue-white screening technique means that those cells that end up taking up the recombinant plasmid will grow white on selective media that contains ampicillin, IPTG, and xgal. Those that contain this original vector will go blue on that media because the LAC-Z gene is still intact, and that makes beta-galactosidase, which splits the xgal into that blue dye color. So what we want to do is put our Arabidopsis DNA somewhere in this LAC-Z gene so that we disrupt the LAC-Z gene so our recombinant cells will grow white. So we need to decide which enzymes to use. The trick is we only want to use enzymes that are found in the polylinker. We don't want those enzymes to be also at other places, and we need enzymes that only cut the polylinker one time. And those enzymes that we use also have to, of course, be found in the Arabidopsis, so that when we cut both molecules by the enzyme, we'll generate sticky ends that will stick back together to link the two molecules of DNA together. So I don't want to use SPE1 because it's found multiple times and it's not that are outside the polylinker. I don't want to use DRD1 because it's also found somewhere outside the polylinker. EcoR1, though, is only found one time, and it's present over here in our Arabidopsis DNA. So EcoR1 is a good choice. And I'm just going to put a little arrow next to both of my EcoR1s, one in the polylinker and one in the Arabidopsis. We've already said we don't want to use SPE1 or DRD1. RSA1. Let's see. RSA1, it's only found right here in the polylinker. Oh, good. It is found in the Arabidopsis. So we can use that. When I look for BAMH1, I see it's not found in the Arabidopsis. So the only combination we can use is EcoR1 and RSA1. So those will be my two enzymes. So what is the net gain in size? When we cut this plasmid with the two enzymes, we'll lose this little piece of DNA between the two enzyme cuts. RSA1 cut at base pair 365, and EcoR1 cut at base 200. So if we get the difference between those, we'll have the number of base pairs that we lose. And that is 165 base pairs lost. The amount added was the distance between EcoR1 and RSA. Now, the numbers on my map here are the base pair at which the enzyme cut. The numbers on my map here are the number of base pairs between the enzyme cut sites. So 500 plus 1350 is the amount added, and that will be 1,850 added. Our net gain, which is what I need here, is what we added minus what we lost. Whoa. So 1850 minus 165, and that's going to be 1,685 base pairs. What's the size of our recombinant plasmid? The size right here is my initial plasmid size, 3,100 base pairs. And then I added some and lost some, so I have a net gain of 1685. So my final size is the original 3,100 plus my net gain, 1685. And that's going to be 4,785. Okay. Now this last one is going to take a little bit longer. It says, draw the map of the recombinant plasmid. Indicate the cut site for each enzyme and the base pair where each enzyme cut. So let's see if I can slide this down. There we go. I think I have room to write here. We know our recombinant plasmid will be a circle because it's a recombinant plasmid. Base pair number 1 is still up right at the top. Now, we didn't change anything until we got to the EcoR1 site. So we still have an SPE1 site, and that's at base pair 75. And our next site is the EcoR1. And that base pair is still at 200 because we didn't change anything up to that cut site. Now, that sticky end from EcoR1 stuck with the sticky ends up here for EcoR1. So our next enzyme cut site will be 500 further down, and it will be SPE1. 200 plus 500 is 700. So this SPE1 is at 700. Now we're still adding some of this Arabidopsis. Our next enzyme cut site is RSA1. And it's going to be 1350 after this SPE1 site. So I can calculate that as 700 plus 1350, and that equals 2050. And I'm just going to make a note here. This 700 was calculated as the 200 from the EcoR1 site plus 500. Okay. Now we've glued back this RSA glued to there. So our next enzyme cut site is BAMH1. Originally, BAMH1 cut at base pair number 420. Now it's after this entire piece was added. The number at which BAMH1 cuts will be what it was plus our net gain. So 420 plus the net gain was 1685. And that's 2105. All of these other cut sites around are the number that you see on the original vector map plus the net gain. So next we have SPE1, and it will be at 1260 plus our net gain of 1685, which gives us 2945. And let's see, now we have an origin. This ORI is important. That's the origin of replication, so that the vector can replicate in the bacterial cell. And up here, we're going to have an ampicillin resistance gene. And that ampicillin resistance gene is very important also. It's used as a selectable marker so that only cells that took up a plasmid, whether it was the original or the recombinant, will actually be able to grow on the selective media that contains ampicillin. Our next site is DRD1. It was at 2350. So we have to add that 1685 net gain, and that brings it to 4035. And our last site was an SPE1 also. It started off at 2700. We add the net gain to that, and we get 4385. And then lots of times people will put the total size in the middle of the plasmid, just like I did up here. So remember, we've already calculated that total size. It's 4785. Okay. I hope that helps. I'll leave that complete map up here just for a minute or a few seconds. All right, let's go on to the next problem. The next problem looks at this a bit differently. We're starting off with a gene that's already been inserted. So we have a recombinant plasmid that we're starting off with, and we want to analyze it a bit. It says the early senescence gene from a tomato was inserted using the EcoR1 and RSA1 sites of the polylinker of a plasmid vector. The original vector contained ampicillin resistance and LAC-Z genes with the polylinker located in the LAC-Z gene, and the map is shown here. You want to first find out how big the early senescence gene is, and then you want to cut this recombinant plasmid with the enzyme SPE1 and see how big the pieces would be after they're separated by electrophoresis. So we look at the process here, and it says we use two enzymes, EcoR1 and RSA1. So there's EcoR1, and there's RSA1. Those were the two used as the enzymes that we were cut with when we inserted our insert of the senescence gene. So which way is the senescence gene? Is it here or is it there? Our original vector had to have the ampicillin resistance gene and an origin of replication. So this is going to be the senescence gene, and the part going around this way is the vector, and there'll be a piece of LAC-Z up here and a piece of LAC-Z down there that LAC-Z doesn't work anymore because we stuck a gene in there. So to find out the length of this gene, we take 1650 minus 150. And that's 1500 base pairs. Now we want to cut our plasmid with the SPE1. Let's see if I can find a different color here since I already scribbled on that map. We want to cut at every SPE1. There it is. So here, 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 there. Okay. Now the rule that we go with is the number of cuts that you have on a circular piece of DNA, like a plasmid, is the number of pieces you should see on electrophoresis. So since we cut it 1, 2, 3, 4 times, we should see 4 pieces. Let's see if we can figure out their sizes. Now I'm going to start here and go around. So one piece goes from here to here. 2500 minus 525. It's going to be 1975. And that was 2500 minus 525. So that's that distance. Now the next one, 3250 minus 2500. So going from here to here, I've got 750. Our next one is 4950 minus 3250. And when I do that math out, there to there gives me right at 1700. And now this one I can't just do by simple subtraction because I'm going across the number one base pair. This is like saying it's 10 till 12 until 5 after 12. How many minutes is that? We have 10 minutes here plus another 5, it's 15 minutes. So what I would do is I would say our total initially was 5,500 minus 4950, and that's going to be 550. So that's a little piece here is 550. And then I've got another 150 from there to there. And oops, not 150, all the way down to here. We got to go this way, not down to there. There we go. So scrap that. We've got from here to here is 525. So I've got a piece there and another piece there. So I'll add 525 to it. And it looks like 1075. So from here all the way around to there to get all the way to there. Now I have to rank my pieces like I would see them on a gel. In a gel, the big pieces don't go very far and the little pieces migrate really far. And that's because these pieces are not floating on top like ice skating. They're actually going through a gel maze. And a big piece struggles to go through that maze quickly. So my biggest number that I had, let's see, I've got a 1975, 1700, 1075, and 750. So 1975 is the biggest. If I was looking at my gel, there's the well. 1975 is the biggest. And let's see, 1700 is next. 1075 is the third one, and 750 is the smallest. So this is what my gel would look like. And these are the 4 pieces I would see going from top to bottom, where the top was the well where the sample was loaded on the gel. Okay, enough of manipulating recombinant DNA for a bit. Let's try something a little bit different. Here, we are trying to figure out who are the parents for each of the puppies. We have 6 adult dogs, 3 males, 3 females. Each of the female dogs had 1 puppy. So we've got a total of 3 puppies. We don't know if each male fathered a puppy or not. So we want to determine which adult dogs are the parents of each puppy. And we amplified the same STR region in all 9 animals, separated them on a gel, and here are our results. So I would tackle this by just starting at the top and listing out who are the possible parents for each puppy. So puppy 1, and look across here, mom A, mom B, mom C. None of those moms have that band. So this band must have come from the dad, and the only dad that had it was dad C. So I know that puppy had dad C. That means this band must have come from the mom. And let's see. Well, that's easy. Mom B is the only mom that had that band. So one thing that you may find helpful is to use a piece of your scrap paper to help you track these lines across so we can see the only band that was in common with pup 1's top band was dad C, and the only one here that was a mom was mom B. Okay, let's go to pup number 2. So pup number 2, okay, well, pup number 2, we could have mom A giving that top band, or we could have dad A giving the top band. And let's see the lower band. Oh, there's a bunch of them. It could be. It could be mom B, mom C, dad A, or dad B. So let's think of the possible combinations. If it was mom A, it could be either dad A or dad B. So possible combinations. So number 2, mom A, because we'd have to have, if the top band came from mom, the lower one had to come from dad, dad A or B. Or we could have mom B. Oh, nope, can't have mom B. Look at that. We already used mom B, and we know this had to be the parents of pup 1, and each mom only had 1 pup. So that's not going to be it. Or it could be mom C and dad A. All right, let's look at pup 3. Pup 3, well, pup 3, that top band is mom A, no other buts. So we know now that number 2 cannot be mom A. Well, let's see who's pup 3's daddy is. And it looks like the only possible dad is dad B. And going back here, now our only choice for pup number 2 is mom C and dad A. Okay. I hope that you found this helpful. Thank you for your time and attention, and I hope you have a wonderful day. Please let me know if you have any questions.